So the bit on the left is actually showing what's going on in the mouse's hippocampus,
In the hippocampus.
as it's moving around
As it's moving
in
around.
real time.
Yes.
It's just mind-blowing. So the hippocampus is... How big is the lens on the camera? It must be like... You finished your PhD end of last year, right? And you awarded it fairly recently.
Yeah, it was about 3 months ago.
And I read it, and I was really surprised actually. It was much better than I thought it would be. Yeah, because you did it so quickly. Like, how long did it take you?
3 years.
3 years in total,
Exactly.
which is like twice as quick as normal, right?
I think here in Spain, 4 years is the normal
Right.
period of a
Yeah,
PhD, yes. In Europe generally.
Yeah.
In the US it's 5 years always,
Yeah.
at least. I was actually lucky that when I joined the lab the data was collected already.
Right.
Although we had to repeat it for some reasons that I may tell you later. But when I were there, if I wanted to do all the experiments on my own, it couldn't finish in 3 years, yes.
Right. Yeah.
It was
Yeah.
shorter than that.
Yeah. So what really amazed me about it was the technology that you use is really, really cool. So what I thought is if we can go through and you can just tell me a bit
Sure.
about it, and I've put some slides together here.
Anti-NMDAR Encephalitis. This is an autoimmune disorder that was discovered in autoimmune disorder.
Yep.
Yeah. So NMDAs are the receptors. NMDAR is the R for receptor. So this is a receptor which is found all over the brain on the cell bodies, and anti-NMDAR is an antibody which attack these receptors.
Right.
Internalize them. They are on the cell body, so these antibodies cause them to internalize. Actually, they disappear and they don't function.
Right. So we're talking about neurons.
Neurons.
At the end, they have synapses that like touch together, and these NMDR receptors are like in the ends of the synapses. Is that right?
Exactly.
Yeah. And so they facilitate the communication between the two cells.
Yes. They are the receptors. When they open, the current passes through the cell and the spike is produced.
Yeah.
So the disease, let's first talk about the disease.
So the disease causes antibodies that attack these receptors.
The disease is caused by the antibodies,
Yes.
actually.
Yeah.
Yes.
So
there are these antibodies which attack these receptors. These receptors are mostly found in the hippocampus.
right.
They're all over the brain,
Yeah.
but mostly concentrated in the hippocampus.
So the hippocampus is like right in the middle of the brain, isn't
Yes,
it?
it's like in the deep into the medial temporal area. It's very small.
It's very small, right?
Yes.
And it's thought that it's important for memory formation.
Yes.
Right.
Exactly.
So all kind of memories go through that thing.
Well, if you don't have it, the main findings about the memory was all related first to the medial temporal lobe. No one knew what exactly here, but then they realized that if you remove this hippocampus, you're not able to form new memories.
Right.
You probably don't lose the memories that you had previously,
Yeah.
but you're not able to form memories.
Right. So in this case with anti-NMDAR,
NMDA.
those receptors are gone. That means the neurons in the hippocampus can't communicate
Exactly.
properly, and so people who have this disease can't form memories, right?
Yes. There are many different symptoms. Maybe we can go back to the previous slide
Sure.
to look at the different phases of the disease. It starts with some psychiatric symptoms, but very soon rapidly evolves into a deep, to an acute phase where people have problems with movements, they may have seizures, they
Right.
lose consciousness,
Yeah.
sometimes even they go to coma,
Yeah.
but it doesn't last more than a few weeks.
Yeah.
Normally treatments are helpful.
And it's fairly rare. So
It's not fairly rare actually.
Is it not?
This is the most common encephalitis in the world.
right, but in terms of the number of people
I don't know.
got
no,
it?
I don't remember.
I think from your PhD, I kind of made a guess that in the United States there might be 1,000 people or something like that. You know, it's that kind of level, right?
Mm-hmm. Well, among encephalitis, I know that this one is the most common one. I wanted to tell you about these different phases of the disease,
Yeah.
where the people go to this acute phase, they go to coma, they need intensive care, but after they recover, they go back to their normal life, but the recovery period lasts so long, even up to 5 to 7 years recently is reported,
Right.
that they have problems with their cognitive functions. They're living their life, no problem, but they can't form memories. They don't have working memory. They usually forget the very recent things that they've learned. Their attention has problem,
Right.
things like that.
Yeah.
And there is no reason for it, because they're neurologically they're recovered. There are no antibodies anymore,
Right.
and there is no explanation why this recovery is lasting so long. That was the main question. We wanted to know what is making this recovery period lasting long.
Yeah. Yeah. So the technology, I think, is just mind-blowing. So I did a biology degree 30 years ago, and when it came to neuroscience, you know, recording the activity of neurons, what I remember is that they stuff like they had to use squid cells because they have particularly large axons, and you can stick a needle actually in the cell to record it. So the way this technology has progressed, I just found is amazing. So you use mice, and in biology there are certain creatures that are used a lot, which means they're very, very highly studied, so they're called models, and the mouse is a model, so we know lots and lots about mice, right? And one of the amazing things, of course, we're both mammals, and so a lot of the kind of low-level biochemistry is not dissimilar. And in this case, the way the mouse brain works is the same, more or less.
Especially in the hippocampus, yes.
Yeah.
Hippocampus is important for memory, first of all,
Yeah.
and also for navigation and space recognition.
Yeah.
And it has been many times repeated, the experiments showing that this same brain region in the mice has the same role as in humans.
Yeah.
So without hippocampus, they're not able to find their ways at solving mazes, for example, or navigating properly.
Yeah. So in humans, the hippocampus is small, so in mice it's teeny weeny, right? And I think we should say that this kind of thing is not done lightly. You know, you have to go through ethics committee to have it approved, and then you have to follow procedures with the mice. So it's you know, using mice in these experiments is taken very seriously, right? So this is just amazing. But you take spinal fluid from the people with these antibodies, and you inject them into the relevant part of the mouse.
Exactly.
So you need to inject some mice with fluid from people with the antibodies and some mice with fluid
Without
the
antibodies.
antibodies. So those mice are your control mice, and then you have the because you need to compare the difference between having the antibodies and not having the antibodies, and everything else has to be exactly the same, right? So you have control.
So we do it with the patient. We take the fluid from people who are suspicious for anti-NMDAR encephalitis, and then the fluid is tested whether they contain antibodies or not. Those who have the antibodies are used for the experimental group of the animals, and those who have other disease without antibodies, we use them to inject to the control group of animals.
Right, yeah. And then this gets even cooler, right? Because to detect what's going on in the brain, to see how the neurons are firing, my understanding is you inject the mice with viruses they're phage viruses, are they? The ones that kind of inject the DNA into the cells. And those viruses have been genetically modified so that they attach to specific neurons, is that right?
Exactly.
And they've been genetically modified so that they inject a piece of DNA that encodes for a protein which fluoresces, right?
Exactly.
It's just so cool.
So this GCAMP6M, this is actually a calcium indicator.
So it fluoresces when there are calcium ions, and the calcium, because of the way neurons work, when they fire, calcium ions go up, so the cells glow when they fire, right?
Yes,
it's
that's
just
true.
Amazing, right?
Yes, that is a very big discovery in neuroscience actually. It's a very recent thing. In 2001 it was discovered, the GCAMP6M as a sensor. It's a genetically encoded calcium sensor, and as you said, when the cells are active there is more calcium, and if this is expressed genetically in these cells, as soon as they become active, the fluorescent is shown. There's a confirmation.
Which is just amazing, because the neurons are tiny, 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 weeny things, right? And it's a real mess in the brain. It's like kind of blancmange with like, it's just messy and really, really small. And with this technology, you can actually see what's going on.
The big thing is that you can see the population activity. But with electrophysiology, you could maximum look at and measure tens of cells.
Yes.
But with calcium imaging, you can see hundreds of cells all at the same time, and you can even track them over time. That is the most amazing thing about it, because when you put needles in the animal's brain, you're not able to find the same cell tomorrow or even an hour later. But with this technology, you see hundreds of cells, the same cells tomorrow, and you can track their activity across a very long time.
So sticking needles in was one of the old techniques, like I described with the squid cell. But even things like fMRI, where you thought, can you get fMRI for mice?
Yeah, you can actually.
Right?
You can, but the resolution is not as good.
Yes. So I was going to say, so the resolution of fMRI is like a cube of sort of a couple of millimeters on each side, and so within that size, even though it's tiny weeny to us, there'll be maybe 1 million neuron cells, something like that. So we've gone from that kind of resolution to being able to see individual cells.
Yes.
Right?
Well, they have different usage. You want to know which part of the brain is active, fMRI functions perfectly. But if you want to do what each individual neuron is doing, which is very difficult to analyze and understand when you look at a lot of cells becoming active in a very random pattern, it's very more difficult to analyze it. But yes, it has its different usage.
So we got a little bit of video on this one. Here we go. So this is video you've taken, is it?
Yes. This is my mice.
What's going on here?
Well, the mouse is moving around, doing a task that I can explain to you. There are two dishes there. One of them has a small food pellet in it. The mouse is supposed to find that, and after several times coming to the same box, going directly, like it goes navigate the environment, and then it learns the correct location of the food pellet which is in the dish, and almost after 5 days of practice, the mouse goes directly to that correct location. So we wanted to look at the hippocampal activity when the mouse was performing this task, and at the same time there is this mini microscope which is mounted on its head when it's moving. So on the right you see these little cells. So those are the cells which are becoming active, and the fluorescent is shown when they're active. So you can see at each moment of the video, the behavioral video, which cells are active.
So the bit on the left is actually showing what's going on in the mouse's hippocampus,
in the hippocampus
as it's moving around, in real time.
Yes. So what I wanted to do was to find out how these activities of neurons are related to the locations that the animal is. So I could compare them.
It's just mind-blowing. So the hippocampus is, how big is the lens on the camera? It must be like...
The lens is the lens is kind of a tube which we implanted. It's in like micrometers, very small.
Amazing.
But the mini microscope, which is on the head, is in like maybe 1 centimeter.
Yeah. So that's not all the core technology you're doing. So you've got a camera on the mouse, of course, recording them moving around, and then you process that using AI technology that basically digitizes the direction,
the
position,
paths, the trajectories.
What is that called?
Deep lab cut.
Deep lab cut.
That's also a very new technique.
Yeah.
Before, I did try to do it on my own, to track the animal, to find this shape of the animal in the video and track it. But this is a deep learning toolbox.
I actually saw there's a website where they compete to make the most efficient models. So they put up data and then, it's very cool.
So you have to first, as all the machine learning techniques and deep learning techniques, you need to first give it a sample data to train with. So you manually label the animal's body parts that are here shown, like those dots that you see are the body parts which are important for the model to learn the shape of the animal. And then it starts looking for these points in all the frames of the video, and then finally you have very precise trajectory of the movement.
Yeah. So the mouse actually has to do, right? So you have a box, and you, over a series of days, you put the mouse in the box, and there's always food in a particular location in the box. And so the first day, so the image on the left here shows maybe the first day, and the mouse is just exploring. But after a few days, it just knows, oh, the food's over there, and goes straight there. So this is an example of a memory of a place.
Location.
Right.
Exactly.
So let's go on to this next one. So what are we seeing here? So we've got day -1, day 0, day 23, day 41. So
well, day 0 is the day that I started injecting the antibodies to the animal.
Right.
So before starting the injection, we pre-trained the animals. We wanted them to learn this location, these environments. They were 2 different boxes, 2 different food pallets, 2 different locations. We wanted them to learn the environments, to be able to distinguish these two, and whenever you put the animal in each of these boxes, the animal knows exactly to what direction it has to go. And then when the memory is formed, we started on day 0 to inject the antibodies, and then we wanted to look at their performance in finding the correct location, if they are losing the memory that they formed or it is not affected.
So the control ones presumably don't lose the memory, and the ones with the antibodies injected lose the memory progressively, do they? Right.
Exactly. So in day -1, when they're still learning, you see that it takes a long time navigating until it finds the pellet. But then on day 0, it has, that is -3, I think, yes? Yeah, because on day -2, okay. On day 0, the animal goes directly there. And then it's even doing better in day 22. When there is a delay, a longer gap between the experimental sessions, they may get confused and spend some longer time looking.
And of course, all mice are different, presumably. This is just one mouse. So you took data from various mice, got the kind of average response, right? So describe what this slide is showing.
Well, this is a place cell. The place cells are the typical cells in the hippocampus, which are responsive to special locations. So mice, humans also, have many cells in the hippocampus where each of them is sensitive to a particular location of each environment that you learn. Like there is one cell that whenever in this room you come to this location, it starts firing. And then you go to the other locations of the environment, it keeps silence. The same cell, when you go to another environment, to another room, has a different place field. The location that the cell is responsive to, is sensitive to, we call a place field. And then these place cells have memory. They are actually hypothesized to be the neural basis of memory, because this cell, which is responding to this specific location, when you come tomorrow again here, it again responds to this location, and a week later the same. So it seems that the cell has a memory for this particular location.
Right.
And episodic memory is very much related and dependent on the spatial information. So this is an example of a place cell.
Yeah. So on the left, that's another diagram of mouse movement.
Yes.
And then the little red dots on that are where a particular place cell is firing.
This is just a particular. It's just one cell here.
Yeah.
So
and then the
red
two
dots,
things on the right, that's data from the camera that's been processed to get those are kind of averages of firing, are they,
exactly,
for that particular?
Exactly.
So neurons tend to like fire a frequency, right? And so
the higher the frequency is, the more responding the neuron.
So the yellow dot would be when that place cell is on average firing.
Yes, that's
at a high
exactly the
frequency.
place field. The yellow area is the environment, the location where this cell is responding to. And if you compare it with that behavioral figure on the left, you can see that these small red dots are the spikes. Each dot is one spike.
Yeah.
So you can see that in the same location of the environment, the cell is spiking, and that's just the same thing in the domain of firing rate.
So one of the things I found interesting in your PhD was that if we have the box with the mouse moving around in it, then there will be particular place cells that fire in precise locations, and then there'll be other ones that fire, for instance, along this wall, or like along these two walls, or that they fire in particular circumstances. And so at any one point, more than one place cell can be firing, right?
Of course, yes.
And the way I think about it is that so what you've got there is an encoding, a way of encoding locations. So as it moves around, the different locations have like different postcodes, right? Depending on which place cells are firing, right? And the way I think of it, it doesn't really matter if you start off with the mechanism being kind of random. So you have inputs from the mouse's senses, so its eyes, obviously, its whiskers, its little feet. They're very sensitive in mice, aren't they?
They are.
So all of that data's going in, and you could have it working completely randomly, and you would still get those. You don't need a sophisticated wiring model in there. If you start off with things just all linked up together randomly, then you'll get that postcode in different locations, right?
That's true.
Which is super interesting.
Yes. That's a map, actually. They call it a cognitive map.
Yeah.
That you have a map in your brain from the locations which are not necessarily continuously connected to each other as the real world is, but the map has its own code for recognizing the environment.
Yeah.
Yes, that's true.
Because it made me think I think it's in The Blank Slate by Pinker. He says he asks a really interesting question about twin studies. Of course, there are lots of twin studies because they're genetically identical, and of course, if you get genetically identical twins growing up in the same environment, then if you think about it very basically, you might expect them to be identical in their behaviour. But Pinker says people tend to focus on the things that are the same, but what's really interesting is how much is not the same, right? And his explanation for this was that basically when our brain is wiring, the initial wiring has to be random to an extent, because there's just not enough data within the DNA to be able to store the amount of connections that you have in the brain. So you get random configuration, and then your behavior derives from that. And I just, when you think about these things, you realise, yeah, it can actually work with random setup, this kind of thing with memory. So
maybe I can add something to your point, that if you look at the first random place cells which are in the hippocampus in a typical environment, and then you put some rewards, let's say some interesting objects, then more and more cells become responsive to the things in the environment.
Right. Yeah.
So there are some cells which start becoming more sensitive to the borders, let's say to the walls, to the objects, or to directions which lead the animal to a food reward, let's say.
Yeah.
So this is your environment, your interaction with the world, which is making the random configuration, the random setup of this population of cells to form a map.
Yeah. Would it be right in thinking, I mean, we don't really understand exactly how memory works, right? And this kind of work is contributing to that understanding. And the way I kind of imagine it working is that the hippocampus kind of provides the postcodes, provides the lookup data for the memory itself, but the memory itself isn't necessarily stored there. I would imagine that it is encoded somewhere else in the brain, and there's all the stuff that happens when we sleep, when there seems to be some kind of memory consolidation. And I would imagine that the hippocampus is essentially providing the lookup data for that, and it's actually going to.
Yeah, that's
is that how it works?
That's true.
Well,
Yeah. We don't know how exactly the information is sent from the hippocampus to spread all over the brain, to form this spreaded kind of memory. But we know that NMDA specifically is the main machinery, the brain machinery mechanism for long-term potentiation, which is the neural basis of memory formation. Long-term potentiation is when the connection between two neurons becomes stronger.
Yeah.
And to have more receptors on the cell body to receive more inputs, to become to tie together the cells, you need this NMDA to function. And these are functioning in the hippocampus. When these connections are strong enough, then the memory is sent all over the brain.
Yeah.
Well, we don't know the mechanism of this yet, but we're looking for it.
Yeah. It's super fascinating. The other thing that it strikes me is that it seems that there are particular ways of encoding memories, right? So you have location-based encoding, what this is talking about, and you also have encoding related to time, right? Is that how it works? No?
Time encoding?
Yeah, I don't know. It is. Do you have an encoding where, when you think, what I did last Thursday or whatever, there's obviously some kind of lookup going on in my brain that isn't place-based, right? Because I'm not trying to access it via that.
Yes. We know that the time is also encoded in the medial temporal lobe.
Right.
And there are cells which encode specifically time.
Yeah.
I don't know how the receptive fields work.
We know that place-based encoding is powerful, right?
Yeah, let me tell you one thing. There are some of these place cells which encode not only the place but also time.
Right.
For example, the place cell, which is responsive to this location, you put it today, it increases the firing rate, let's say, to 100 spikes per second. And then the next day, when the time is changed, it is still responding to the same location, but with a different firing rate, let's say 50 hertz. And then if you add a context to this environment, for example, there is one context that there is, let's say, a music playing, and when this music is playing, you have to do a certain task, the same environment, another music, another task. these cells start to distinguish the conditions, the timing, but responding to the same location just by changing the firing rate.
Yeah, that's fascinating, isn't it? And knowing how memories are encoded we can use this knowledge to improve our own memories. I'm just thinking another thing that I thought of was that we've known for a long, long time that one of the most powerful ways to memorize things is through location. You've heard about memory palaces,
yes.
right? Do you know about this? So, it's one of the ancient Romans, it's probably Cicero or something like that, wrote there's a story of ancient Greeks, right, going to a dinner party, and the ceiling collapsed, and fortunately he'd just gone out for a smoke or something. The ceiling collapsed, and he could actually remember everybody. I guess when he was speaking to the authorities afterwards, he could remember everybody there, and he realised it was because it was location based, because he would think about the tables and then he would go around the tables. And in medieval times, they because there weren't printed books, if you went to a monastery or whatever and you knew it had a particular book, you could borrow the book for a couple of days and then you'd have to give it back again. So you actually had to memorize as much as you can. And they used to use this memory palace technique, where you would imagine a physical space and you'd move through the physical space in your brain and attach the information to those physical locations.
Exactly,
right?
That's what I did when I used to study at school.
Yeah.
I think that's very reasonable that we have evolved our spatial memory because we needed much more than the conceptual memories, because we needed it more to survive. And now when you want to memorize something which is not related to the space, you just put it, when I want to memorize a piece of information, I try to put it in my mind on the cabinet in my kitchen, and the other one in the shelf in that room. And whenever I want to
when you're
recover
doing
it,
that,
I just go to the room, I pick it up from the shelf, and I find it.
Yeah, it's great, isn't it? So when you're doing that, when you're imagining moving around in that space, you'll be triggering the place cells.
Yes,
in
exactly.
your hippocampus, right?
Yes. Even when you are not in an environment and you just start thinking and recovering the memories of a location that you experienced previously, the same place cells become active. And that's exactly why another reason that we believe that the place cells are the basis of memory.
This has been so interesting, and there's lots more I want to talk to you about, so we'll do this again soon.
Sure.
Thank you very much, Pasha.
Thank you
That's
for
great.
inviting me. Merci.